Hello, welcome to the October 2015 Getting Started with iPlant webinar. I'm Jason Williams, coming to you from Cold Spring Harbor Laboratory in New York. Welcome. This webinar is an invitation we extend each month to the 500 or so, this month I think about almost 800 new users that signed up for an iPlant account. And the purpose of this webinar is to try to give you a little bit of an overview of all the things that iPlant has to offer to advance your science, your research, your education. And what we'd like to do over the course of the next hour is give you an introduction to several of the most popular platforms, answer your questions, do a few demos, and really give you a way to get started with iPlant. We know that and even as I look on the survey, the way that many people find out about iPlant is by word of mouth. One of your colleagues is using it, probably introducing it. Perhaps you're taking a class that's using it. And so that may not be the full introduction. And so what we hope to give you an introduction to today here is some of the broader aspects of what you have to look forward to using within iPlant as a platform. So before I go into all of that, what I want to first introduce you to is the technology that we're using for this Adobe Connect webinar. First, at the bottom of the screen there, you'll see a place where you can actually ask some questions. And off to the side here, I can actually monitor what's going on and take a look and see what questions people have asked me. So at any time, if you do have any questions throughout the webinar, that's kind of the point. You can always watch a video, and of course, actually this webinar will be posted to YouTube very shortly after, so that if there's anything you missed, or if you want to share it with somebody else, you're welcome to do that. But we really encourage you, if you have questions, go right ahead and ask them at the bottom of the screen. At any time, I'd be more than happy to take those questions. At the top of the screen, you're also going to see a little icon, a button with 4 arrows in it, where you can actually make the video full screen. And when I do some of the hands-on demos and take you through some of the platforms and products, it might be useful to make the video full screen at that point so that you can see it in a little bit more detail. On the side of the screen, one of the things that you could see, I think, are some downloads, including this presentation, some useful links that will help you get started. So with that, what I'd like to do is begin the part of my presentation that has to do with a little bit of the history of the project. We feel it's really important for you to know what iPlant as a project is, how we got started, where we're going, and if you know something about that, then certainly you can have an idea of how you can contribute and participate. And then we'll go on to a couple of live demos of some of the platforms that are your most popular ones, the ones you're most likely to use. Okay, so let's get started. iPlant as a project actually started with a little bit of a vision. And being from New York, perhaps, I took this portmanteau, the New Yorker cartoon on the left-hand side there, and also an image that I think most biologists are familiar with, the image of the double helix. The New Yorker cartoon is actually a little bit historical. In New York, there's the Hudson and the East River, and this particular person being depicted here, I believe, is Verrazzano, an Italian explorer. And he's saying, someday this will all be infrastructure. So what's the point here? Well, in New Yorker cartoons, sometimes it's a little bit hard to grasp. But biologically, I've placed it next to the double helix because the question that I think everybody in the project, every biologist who's concerned with this is thinking about, how do we think about what biology will be in the future? And in fact, as far as iPlant is concerned, what will this have to do with infrastructure? Well, as you probably know, when we're thinking about biology today, it's very much different than what it was, let's say, back in the 50s when the double helix was discovered. Now, being a biologist often means not just working in the lab or the field with test tubes and pipettes, but actually sitting at the computer to do computational biology. So if you look back at the image here, one of the things that we felt was really important in biology was actually to build infrastructure. Now, in the case of a city, it's the roads, the bridges, the tunnels that get us where we want to go. And as you'll see as I go through this really short talk on the history of iPlant, the infrastructure that we've been building is cyber infrastructure. In fact, that's our vision, to enable life science researchers and educators to use and also extend our foundational cyber infrastructure to understand and ultimately predict the complexity of biological systems and the dynamic nature under environmental conditions. That's a bit of a mouthful, but essentially what it means is we want to enable scientists to ask really big questions, and in fact we believe that cyber infrastructure is the way to do that. Now, what is cyber infrastructure? Well, at one level, cyber infrastructure is no different than what you've already been using, perhaps in your daily lives, perhaps in your research. It's data storage, it's software, high performance computing, and also, and especially in my opinion, people. But it's organizing these things into systems that can allow us as biologists to solve problems of size and scope that otherwise would not be solvable. So in other words, we're organizing these different systems in a way that's custom tailored to the types of questions that biologists, whether you're working in molecular genetics or whether you're working in population ecology in the field, we think that all of these systems are key to being able to ask big questions. So there are a lot of products within iPlant as a project. Some of them you may be familiar with, maybe even got a chance to use so far, but some you may not have ever even realized existed. So that's again the point of this webinar. At the top level, there are the everyday sort of consumer-facing, if you will, products. The Atmosphere Cloud Compute, the Discovery Environment for your web analyses, but underneath all of those things, and we'll cover them to some level of detail throughout this presentation, there are other things that are perhaps hidden. One of them that's certainly quite important is the data storage, but that's just as important as the authorization and search, being able to search the data, being able to know that your data is secure. Underneath that are some of the more technical components, how we share data across our systems and with you, how we set up a cloud that anyone can access, and then actually the physical systems from our computers based at various locations that give you access to HPC, to doing the cloud, to working with databases. So all of these different products in this sort of layer cake model contribute to the cyber infrastructure that we've built. If you want a different, maybe even in some ways more practical take on it, really what you deliver and what you get with cyber infrastructure is platforms, tools, data sets, storage and compute, and that people layer, the sort of training and support. And today is an example of that. We're really committed to making sure that our users have a place to get support for their questions, for their science questions, for their technical problems. All of those different things are part of it. Another important question about iPlant is what type of problems can iPlant solve? So, I mean, I don't know if everyone on the webinar is a plant biologist, but certainly the name implies plants. But really, iPlant services needs of all sorts of communities. So certainly we started out with crops and model plant systems, but also non-model plants, also animals, microbes. So really, if you want to summarize it, iPlant is basically built for data. Any type of biological question, problem that you're working with, from a breast cancer paper that we had recently, one of our users told us about, to working with your standard Arabidopsis, iPlant is built for data, and so as a biologist, you and your colleagues can really use it and not feel that it's just constrained to simply one domain of science, and we'll see the breadth of it a little bit, I think. How was iPlant built? So going back to the history, and I'm always through my short talk, but I do believe it's important that you have a little bit of an idea. iPlant was built sort of at the time when I think most biologists certainly and other sciences may have already been doing this for some time, but we realized that data and so-called big data was really important to science. But one thing that we did know from the very beginning of conceiving in this project is that it was always going to be a community effort. It was never going to be a few people in a room who could really know what you in your domain of science really needs. We had to be more general than that, and we had to really have community input. And so, in fact, more than 1.5 years at the onset of community input, and then even to this day, continual community feedback, we identified a whole landscape of priorities that make a lot of sense. Several years ago they made sense, and they still make sense. The big driver for big data in biology is certainly genomics. And so when you come to iPlant, you'll find tools for reference-guided assembly, de novo, RNA-seq, variant calling, this whole suite of tools that you'd expect to be able to look at genomics with. But one thing that distinguishes iPlant is that we've never been just about genomic data. So environmental data sets, phenotype data sets, all of those things are going to be useful to you when you want to ask, remember I said, the so-called big questions. So being able to do association studies, evolutionary, ecological models, looking at pathways, whether they're metabolic pathways, gene pathways, ecological, all of those types of things require synthesizing different data sets with different data types, different ways to organizing them, different tools, but all that's possible with an iPlant because we've always been concerned about support for all of these data sets and communities and domains. If you look, you can always see this is sort of a little bit of a model. The updated version is always on our website of our priorities and where we are. In a lot of places in genomics, we think that we have really laid the foundation there. Of course, there's always new tools, but we think you have what you need, certainly to get started and also to expand. But also in the environmental products and phenotypic products, we have lots of products there in place, in planning and progress. And so you can always communicate with us or check our website or newsletter and really see what are the new tools that month, what are the goals that we have, what's coming out on a quarter by quarter basis. Another thing that's important about iPlant is that we collaborate. iPlant is not and never had the intention of being the only solution that's possible. We are not there in the business of saying that we can provide everything that you may need as a biologist, but what we do is make it easy to collaborate. So we collaborate. These are just some of the organizations, projects, groups that we collaborate with that certainly have similar aims, but in general, we don't believe that our users should be locked into using iPlant. We want you always to use iPlant when and where it makes sense, and then when the solution exists elsewhere, we want to make it very easy for you to collaborate so that we maybe have shared tools, shared data sets between those different projects, because as you may or may not know, depending on how far along you are, moving data sets is quite costly in terms of the time, the effort, and then not to mention the attention to detail, such as was the data transformed in any way? Did we capture that metadata? Are we sure about the integrity of our data sets as they go from platform to platform? So being able to work within a unified platform that natively supports this level of communication, is interested in collaborating, we hope that that's something that we provide also as our users to a service that makes it easier for you to do your science and not worry so much about, well, this part is in this place and this part is in that place, but really you can do things as more of a cohesive whole. Okay, another important question. Who makes up iPlant? So iPlant is made up actually of several different partners. We're an entirely virtual organization, so quite a number of people are at the University of Arizona in Tucson. Also, we have staff and facilities, and especially some of our HPC systems to exceed at TAC, and Cold Spring Harbor Laboratory here helps guide the education as well as the science, and also at UNC Wilmington. So we are already a virtual organization that depends on cyber infrastructure to function even at that level. We're entirely funded by NSF, and this is really important to recognize because it means that NSF has invested heavily in this cyber infrastructure, and it comes at no direct cost to you as a user. So most of our users are NSF funded in other ways, but you may have other funding sources. You may even be an international user. You're all welcome to use it, but what it really means is, practically speaking, it makes sense to use as much of iPlant as you can because it comes at no direct cost to you. After our renewal in 2013, our special focus was certainly on being able to work with genotype and phenotype science. Our scientific advisory board is one of the primary ways we get feedback from the community as to what we should be doing, and our official scope was certainly moved beyond plants at that time. The sort of proof of concept had been successful, and we really support a diverse user base working beyond plant science. Okay, so that was the history part. I hope it wasn't too long, but I do hope it gives you a little bit of background as a project, what we do. Now let me move into the demos. Before I do that, let me just talk about some key benefits and advantages that we as a project deliver to our users, that we hope we deliver, because these are things important to think about, especially with, and we see this most of the time from our educational webinars and workshops, that many of our users are new to computational projects and they are just getting started with it. So here are some important points to think about and advantages that we want you to realize and even take advantage of when you're using iPlant. So what does it mean to work within a cyber infrastructure, a unified platform? Well, here are 3 points that I pulled out, three broad areas. Number one, get your science done. Most people at the end of the day want to have their scientific product done. They want to have their question answered. And so working with an iPlant, you get the ability to access and manage your data, software to analyze it, computing resources, and even skills and help to use that software and interpret the results. Reproducibility, and this is really key. It's not always obvious at the beginning of a computational project, but you really do need to think about how you're making your science reproducible. And through iPlant, we have a fairly sophisticated way to work with metadata management, to share data and workflows, and we are all about using, and in fact are, an open source project with sustainable tools that are also open source. Productivity is another key factor. So being able to use high performance computing, scalable platforms, what this means is that if you're sitting on a project and an analysis and it's taking weeks to run, may want to rethink that, may want to ask us a question, how can we speed that up, how can we be more efficient? Also really key, how can we be more collaborative? You have the ability to automate and collaborate within iPlant. You can share large data sets instantly with other users. We'll talk about that. And then also, you can spend more of your funding on science and not the software and the hardware. So, what do you get with your iPlant account? Now, even more specific. Well, remember, and here's a nice analogy we like to use, we think of iPlant sometimes as almost the building bricks or the Lego blocks. We have built 80, 85, 95% of what you need. Oftentimes, that last 5% is custom. So our goal has always been to put most, if not all, of the pieces on the table and allow you to build what you need from what's there. If the solution is not available, we'll help you to craft what you need. So thinking about that, here are some of the pieces. On the base, one of the first ones you'll encounter is certainly a data store. So with the data store, you have the ability to share and manage any data set, any files associated with your research. Every user is entitled to a 100 gigabyte allocation. If you need terabytes of data, you may be able to get that. You just need to fill a little request form. The data are automatically backed up, and it's easy to upload, download, and share. And I'm going to do a hands-on demo of that in just a few moments. The discovery environment is a web interface to hundreds of bioinformatics applications. It's a platform that can run almost any type of bioinformatics app. And it's integrated with our data store and high performance computing. And it's also user extensible, even more so now we've added some new features. They may be a little bit advanced, but for people who've not in the coding world yet, but it's becoming even easier to extend by adding your own applications. And of course, you can even request if you need something and you don't see it there. Atmosphere is our cloud compute resource. If you've not used cloud computing, I'm going to show you in a few minutes how to get started with that and why it's a really wonderful product that can make you more productive as a scientist, more effective. We have one-click access to hundreds of virtual machine images. You can fully customize your software. You don't have to worry about other people giving you permissions to install things or worrying with your IT support. You can really do it there. And it's quite powerful, again, because it's integrated with everything. The Agave and Science APIs we will not be showing directly. They are tangible products with Agave. You can get started today, but that's a little bit advanced. However, if you are a little bit advanced, if you're on the sort of bioinformatician, computer science end of the spectrum, you should know about Agave and take a few moments to go look that up or go to the Agave website because with that, it's real science as a service platform. You can define your compute and storage resources, you can mix and match, and you can build your own app store of codes and workflows. So it's really a powerful product. Just we won't cover that one directly today. DNA Subway also as well, we'll not cover in a demo, but it's wonderful for teaching in the classroom. I think of it as classroom-friendly bioinformatics. It's a suite of educational workflows for looking at genomes, DNA barcoding, which has a hands-on lab, as well as RNA-seq, which also has hands-on applications, and it supports inquiry-based experiments for working with students and discovery and publication of data. BISC, we will not have time to cover today, but if you're interested in imaging and phenotyping, this is our solution for working with those data sets. It supports hundreds of different file types, and you can also use and create custom visualization and image handling routines. Okay, that was a lot, but now I'm sort of done giving you sort of a brain dump of information. As I said, this will all be on YouTube, and so you can go back to anything or please share this with your colleagues. But now I'm going to move into the section where we do complete a few hands-on demos. And the idea of these demos is, along with the existing documentation, which I'll point you to, that you can get started. It won't take you through every possible scenario, but we do believe it'll be enough that will help you to take your first steps and feel a little bit confident about them. Let me take a moment just to remind you, at the bottom of the screen again, I haven't seen any questions so far, but if you do have questions at any time, please type in those questions. I'll respond to those immediately, and that will be a way that we can communicate. Okay, so let's move on to our first sort of demo. How do we get data into iPlant? Okay. So just as a recap, iPlant Data Store, you can store any type of file related to your research. So you may have sequence files, you may have movies, you may have documents, PowerPoint presentations, whatever, you can store those within iPlant. You can move those files seamlessly between different iPlant platforms. You could be working in the Agave system or the Discovery environment or the cloud with Atmosphere, and you can move those data sets. You can automate those file transfers, and this is really important. You can share files with your lab members, with collaborators, and even the community at large. Now, for several reasons, there are actually several methods. Our users have several reasons and several use cases for everything from point-and-click interfaces like Cyberduck, the Discovery environment, and iDrop. These are all tools that you could use to move data. And also, many people want a command line interface. They're used to working at the command line. For the purposes of today's demo, we're going to focus on one of the point-and-clicks, the Cyberduck. But let me just give you a little bit of an overview. In the Discovery environment, you can move and transfer files there. This is better for smaller files. You wouldn't want to transfer really, really big things. You want to use one of our other tools. But this is for your everyday things. The Discovery environment is largely like a desktop. You'll see in a moment. So maybe renaming a couple of files or moving them or sharing them. That's mostly what you'll use in the Discovery environment. I'll show you that. Cyberduck is what we're going to do a demo on in just a moment, and that is a really easy way to drag and drop files and folders. If you're used to something like Dropbox, this is basically the equivalent, and you can move very, very large files using Cyberduck quite efficiently. We'll show you that in a moment. iDrop Desktop is another tool. It is available. You can download it. It has some other features with managing some of the sharing and metadata, but some users, this was a little bit more of a complex interface, so it is available, but our preference is for most folks to use Cyberduck. And iCommands. For those who want to use the command line, I'll quickly show you a little bit of that, again, just enough to get you started with that. Okay, so let's give you a demo of Cyberduck and iCommands. This is the point where I mentioned before. You may want to press those little 4 arrows at the top of your screen, and that will allow you to go full screen and see my computer a little bit better. Okay, so let me start Cyberduck here. I've already got this installed, and while this is starting on my computer, because it'll take a moment to connect, I'm going to go ahead and open the iPlant Collaborative website. So at iplantcollaborative.org, this is our website. If you remember nothing, if you don't watch the YouTube video, please remember to come here and go to this, the iplantcollaborative.org website. Down at the bottom in the Learning Center, I'm focused now right in this section called Get Started. And if we skip ahead to this section called Import Data, this is where you can go ahead and download Cyberduck. There's actually 2 things you need to download. You need to download Cyberduck for Windows or Mac, whichever you're using. And then you also need to download something called a Cyberduck profile. So these 2 downloads. Now, you can review the documentation from the link that is there. Open that in a new tab. And this will take you through what I'm not going to show you, which is the step-by-step of setting it up. But it really is not complicated at all. You just download those 2 files, you install it, and then you'll be seeing a window that looks like this. So my iPlant data store is bookmarked here, and I need to double click on that. Now this will probably take, in my experience, it takes about 30 to 60 seconds to mount. But once it does, I'm going to see a listing of all the files and folders in my iPlant data store. And then if I want to move files and folders from my iPlant data store to my computer, then again, as I mentioned, it's very much like Dropbox, where you will move from one side to the other, and it will start the download. So I'm going to give this a moment to start, quickly show you that, and then I'll move on to the iCommands. And again, just reminding you that if you have questions on any of these, you can both chat me now through the bottom, question window, but also again, just to show you and reemphasize, on the iPlant website, down at that Getting Started link, you will find just about all the details of what I'm going to cover today. Let me adjust my size there. And we are in that part called Import Data, and there's a link directly there to downloading Cyberduck. Okay, so on my Cyberduck, which is now connected, I see a whole bunch of files and folders which are in my iPlant data store. I can see the size of those files, when they were modified, etc., folders, etc., all those things. If I want to download something, I can select that folder, and just drag it over to my desktop or wherever on my computer. And then at the bottom there, it will start the download. So I'll see a look at the progress of that particular download. If I want to move something from my local computer, I could take that folder. Let me just create a new folder. Just call it test because I don't want to move a whole bunch of stuff. I happen to be on wireless, which is not the best. I'm going to move this over to my iPlant data store, and it will at the bottom start that. Now these downloads and uploads will take themselves a few seconds, usually my experience anywhere from 10 to 30 seconds to get started, and then they'll start transferring at a pretty reasonable clip. What we've noticed in general is that these things take about 30 minutes to transfer about 100 gigs of data on a really good connection. So you really should be able to transfer large files pretty seamlessly. So that's really it in terms of using Cyberduck. Once you install it, there's not too much for you to do other than connect to your iPlant data store and choose which files you want to move back and forth. So at the beginning, very likely, what you'll want to do is take your sequencing files or your images and start uploading those straight to iPlant. All right, so before now that I'm finished, let's say it that way, with Cyberduck, let me quickly show you, for those who are interested in the command lines, what you would have to do that. Then I'll talk about how do you share that data and actually access it within iPlant, which will bring us to our other demo on actually analyzing that data. So if you are going to use the command line, back at that same place down here, there is a download for iCommands, which you can download Mac, Linux, and you can go ahead and download those things. Once you download them, then you can run them at your computer, at your terminal. I'm going to go ahead and start a terminal on my Mac. iCommands is not, unfortunately, available for Windows, so you'll have to be using a Linux or Mac. And I'm just going to start a window that's a little bit easier to see. There we go. And then the iCommands work very much like Linux commands, except that they have an I in front of them. Now, we didn't create them, despite the name iPlant and iCommands, but I guess at the time in the early 2000s or mid-2000s, I in front of everything was popular. So if I want to see list the contents of my iPlant data store, it's just an ILS, list contents, and that is my iPlant data store. Those are all the directories there. If I want to download something, I can do that. So you would use a command like iget, and I want to download a directory. I want to see a progress. I'll download the VCS file, and I'll do that to my desktop. Okay, and then you could see on the right-hand side of my computer screen there, that folder appeared on my desktop, and on the terminal here, you can see the progress of the download. Now, there are several iCommands. iGet is a download, iPut is an upload, but there are a variety of them. So when you want to see the documentation for iCommands, back here, you can click on the documentation link, and it will take you through it. In fact, there's the iCommand manual that's down there. And you can see the list of commands, how they work, how you set it up. When you first set it up, you're going to have to use the initiate command to go ahead and start logging into your iPlant data store, but this will take you through all of those. Okay, this took no time at all in order to download, but the download is complete and those files are on my computer. Okay, so that's uploading and downloading with iPlant. Let's talk about sharing. In order to share, I'm going to go ahead and log into the discovery environment. I think I have a couple slides, but let's cover just the first few of them, and then we'll go ahead and actually log in, because there are a couple more that we're going to do. Okay. Okay, so in the discovery environment, just going to move over here. As I said, it's a platform that can run almost any type of bioinformatics applications. The most bioinformatics applications are run at the command line, and you can integrate those in the discovery environment. It's integrated with our high-performance computing and data storage, and it's user extensible. The first thing I want to show you in the discovery environment as I take you through login is data. Not only are there some upload and download capabilities, there, but importantly, capabilities for sharing files and folders, even very large data sets. So let's log in now and I'll show you how that works. I'm going to go right back to the iPlant Collaborative homepage. Okay, so this is the iPlant Collaborative homepage, and if I scroll down and I move my mouse over the Discovery Environment section, right away a link appears that says Launch. So I'm going to go ahead and click on Launch, and then I'm going to go ahead and click Login with my iPlant ID. And at this point, you'll log in with your iPlant credentials. And this is the iPlant Discovery environment. And of course, it's opening to things I was at previously. I'm just going to make it a little bit bigger, but remember on your screen, you can definitely make this full screen. All right, so let's talk about data. So if I click on the data window, what I'll see there is a couple of folders, and the one I need to concentrate is the one with my username, which happens to be my last name. And I'll see here all the things that I saw in Cyberduck, all the things that I saw in iCommands. These are my directories, and these are my individual files. I can highlight a file and then go up to edit, and I can rename it. I can move it to a different location from there. I can also move files that I have ownership on by dragging them and dropping them. Now, over here, I don't have too many permissions to move things into, but you can essentially, well, let's do that. Let's expand my directory here, and if I wanted to take this file and move it somewhere else, I could drag and drop. I can, this is a file opening. It's not going to really show me anything, so let's open one. If I want to look at a file, I don't know if I have, well, I can go up to the top and search for FastQ. See what kind of FastQ files do I have in my home directory. Okay. This looks like a FastQ file, and I can click on that, which is something I think you usually don't do on a normal computer, but in the Discovery environment, even large files, this is 130,000 pages, but I can actually look and preview the file and go into it, so that's very nice. There are a whole bunch of other details I could spend some time on, but I want to just focus on data management, and so let's talk about sharing these data. So if I have a file selected, let's go up to something that's not too big. Here's a file here, and if you look on the right-hand side, let's choose one that's actually got something in it. I can see a little bit of data on it, that it's a 1 megabyte file, it's a text file, which that's what a FastQ file is. If I want to share it, I can click on Share, and whoops, let's see. If I want to go ahead and share this file, let me go up to one that I actually own, go back to my home directory. Okay, and refresh that. All right, so if I want to share a particular file, I can go up to Share, and then I have this option here called Create a Public Link, and this allows me to share a file with somebody, almost like a Dropbox, where it's going to create a URL. I click on it, click Create, and it creates this unique URL, and I can copy that link, and if I give that to somebody, open a new window here and click Enter, it will go ahead and start a download for me, just like anything. So you can easily create links to share files with anybody who'd like. Now, this is not, there's two caveats that are important to this. Number one, the files themselves that if you want to share, you wouldn't want to share really, really large files, several gigabytes, because if the download breaks, there's no way for them to recover it. They have to start again. So we recommend that you choose this for smaller files. And also, that link is sort of public. So if you share that URL with somebody else and they post it somewhere or share it with somebody and you didn't intend that, then it's less secure than sharing it in the next way that I'm going to show you. So if I go back to the demo here on my screen, if you want to share the file, if you want to cancel that link, you can come back to a file that's being shared and you can click the little delete link, and that will delete the link. It will no longer work for anybody. Of course, if they've downloaded it, I deleted it twice. If they've downloaded it, there's nothing you can do. But once they've downloaded that link, you can go ahead and delete it. Now, this file sharing only works for individual files. It does not work for entire directories. If you have entire directories, you'll either compress them or do what I'm going to show you next, which is to share an entire directory with another iPlant user. The reason why this is the best way to share is because you can share any size data set instantly. So if I'm going to go ahead and select a folder, I'm not sharing with this anybody. It says begin sharing. So if I was sharing with somebody, it would actually tell me who I'm sharing it with. But I'm just going to go ahead and select it and now select share with collaborators. So the other person you're sharing with needs to have an iPlant account. You can type in their name or their username. When I start typing them in, it's going to search for that user, and I see them in their institution, and I could say, okay, that's who I meant. And then I can assign them permissions. This is covered in the documentation, but a read permission will allow Cornell to see the file. He can even analyze it, but he can't touch it, he can't edit it. I could give write permission, which means that he could write to that file or alter the metadata. And the highest level of permission is own, means you could actually move it. He could even delete it, and I wouldn't be able to do anything unless I had my own copy. So you can decide what you want to do. Now, if I clicked done, this would go through and he would get a notification that this has been shared with him. If you don't want to do that, or if you've shared something with somebody and you want to revoke that permission, you just come back here and then you click that X and it will remove their permissions from them. Now, once I've shared a file, if I go back here on the left-hand side and collapse this under shared with me, that's where those files will be located. So I'll see a folder that has the username of that person who shared something with me. I'll also get a notification up here. So if somebody shared something with me, or if I shared with somebody, I could click that notification. It would direct me to the exact file. But in here are things that are shared with me. Now, you will almost certainly have a number of folders here because there are some files or some directories that are shared with everybody. But essentially, at the end, that's where they end up with. Okay, so I think I showed you now two of the most important basic ways to share files with other users. You can share it with a link, an individual file, or you can share entire directories, hundreds of gigs with another user. And it's never moving those data, so really it's instant. As soon as you give that permission, within a few seconds, the other user will have it. What I didn't really have time to show you here is some of the advanced searching parameters. You can search for your files. Up at the top, I showed you. So I would encourage you to look at the documentation, but you can also really look at the metadata. I'll show you anyway. Take a look quickly. Here, if I have some files and folders there, I also have the ability, let's say for an individual file, let's look at this FASTA file, click it. This FASTA file also has, or can have attached with it, metadata. So if I go up to edit metadata, I could add my own custom metadata by saying, you know, species equals, let's say, A. thaliana. I mean, that could be true. And this is sort of the one-by-one method, but you can go up and actually select templates because we have various things. Everything from, if you're doing a biosample and submitting some data to the SRA, which you can actually do directly through iPlant, you can fill in those metadata templates for your particular samples. But the metadata become powerful also because when you want to search, you can create custom searches. So if I have metadata fields, I could look for something that has the attribute species and has the value thaliana. I could look for things that are owned by a particular user or shared with a particular user or by file size or etc. So that's really important for you to know about, but that's a little bit more advanced. I did want you to know that it's there and it'll make your life a lot easier because at a certain point you won't be able to browse all the contents of your data store. Okay, so that was all that I'm going to show you, I think, right now for data. That's certainly enough to get started. How do you actually analyze those data? So let's do a quick demo of the discovery environment. So back to the slides here. I think we've covered the data aspect of what's available there. There are two other features that are in the discovery environment that we're going to look at. The apps panel, which allows you to actually run your bioinformatics apps, build workflows, integrate new applications. We're also going to take a look at analyses, which is where you can find the status of your jobs, results, you can relaunch jobs, and you can also see a history of the work that you've done. Okay, so we're already in the discovery environment. There's no need to log in again. What we're going to do right now is a simple demonstration of an analysis. And if you get this analysis, then you'll really get how the discovery environment works. If I go to data, I'm going to look at a data set that I have here in community data. And this one is going to be an iPlant training. And DE walkthrough. Okay, so I have here a set of unaligned sequences. Let's see. DE sample plants, excellent. So I have here some unaligned sequences, and I'm just going to use one of the applications to build multiple sequence alignment from this FASTA file. Okay, so I know the data are there. I'm not going to do anything with it. I'm going to click on this next panel called Apps. So this is the catalog of applications within the iPlant discovery environment. There are some features here that I'm not going to have time to show you today. So for example, you can create a new workflow here. You can also, under Apps, request a new tool or create a new tool within the discovery environment if you need to integrate something. I'm going to search for an application. These are all sort of organized according to different topical categories. I'm going to search for an application called Muscle, which will allow me to build a multiple sequence alignment. Now, when I search for an app, or when I'm looking at a listing of apps, there are various things I can do. If I don't know what the app does, I can go ahead and click on the icon, and it will give me a little bit of a short description and a link to a user manual. I can also give that app a rating if I've used it, so I keep track of how that app has worked for me. I can also give comments about it so that people can know if I have a problem with the app. That's one way to do it. But what I'm going to actually do is use the application. And this is, again, any application in the discovery environment is going to work along the same sort of paradigm. So I'm going to go ahead and click on Muscle 3.8.31 and close the apps window. I don't need that now. And my analysis has a name. I'm just going to say, give it an underscore demo. I'm going to give it a comment. This is a demo. And this is really important. So let me just pause for 1 second. Again, when we're trying to do reproducible bioinformatics, it's actually harder to do that in a graphical interface, not the command line, but it's really required. We've baked those things in as you're doing in a very small way right now, but pay attention to that. So always adding comments. If we were in a wet lab and we didn't keep a lab notebook, we wouldn't be there very long. So we've baked into the system, and we hope that you take advantage. Of course, give us feedback of what others you'd like to see, but always keep track of that. That even goes back to the metadata that we were using earlier, and we'll see again a little bit. Okay, so back here, adding some comments. This is just a demo for the webinar. There's an output folder where my results are going to go. In the main, I leave it as default, but let's say you're sharing all of your analyses with your entire lab. You might have a lab folder, and you could change this so that that's your lab folder that everybody's shared with. This application is very simple. It only requires input data and a sequence type, so I'm going to go ahead and browse for that input data. The browse will open up to where I last input something, so I'm going to have to go back here to DE walkthrough. Let's go back to this. DE sample plants. Okay, I'm going to say okay to that, and I need a sequence type. So you've got DNA, and it's a sort of accordion menu. So you just work your way through the app sections, okay? But once you have everything filled out, you go ahead and click Launch Analysis, and you will get a notification at the top of your screen that the analysis has successfully launched. And now you can actually see what's going on with that by going ahead and click on the Analysis panel. And this is, in some ways, a lab notebook because it's a history of all the jobs that I've done and details about those jobs. So if I go ahead and look at the job that I just ran and just select that one and go out to Analyses and say View Parameters, I can see some things. Now, this app wasn't very complicated, but it did have some parameters. It had an input file, and it had a sequence type, value I had to select, whether it's DNA or protein or auto. But all those things are here, including even my original file. So if I click on that link, it'll take me to the original file that I used this with. This is important when you have 30 files and they're all named with alphanumeric strings that may not be human interpretable, and you want to organize and say, what file did I use as input? You can always go back and check. This, of course, I can save as a text file and export that. For more complicated analysis, looks like I did a top hat job. If I go there and view the parameters, you'll see that there's obviously a lot more there, right? Several different file inputs, several different values, but I always know what I did. I never have to be in doubt, and that's all captured as metadata. Okay, now my job status, if I go ahead and refresh this here, I believe I got a notification you probably saw on the side that this is running, so it'll take a few moments. If I wanted to take a job that I'd done previously, but let's say I wanted to tweak it, rather than filling out 20 different parameters again, I could go up to analyses of that highlighted job and click relaunch. The application would open again, except that the difference is this time when the application opens, it will not be sort of a blank slate. It will be populated with all of the different parameters, the files, all the different things that I had used previously. So give that a second to load. Of course, it's taking longer than it should. Let's try that again. And see if it will not take quite as long. I'll put that off to the side and check my status of this other application. Okay. I'm going to try it with a simple app because it's just not, it's taking too long again. But that may be my internet, not you. Okay, so what will happen is that you will go ahead and relaunch the application. It will load with all the parameters that you had previously used, and instead of clicking through everything several times, you can essentially just tweak the one or two parameters you may want to change. Now with here also, if you have a job that's in the running state and you realize, there we go, see, it was telling me that there was an issue there. If you have the app running in a state and there's something wrong with it, you realize you made a mistake, you can also cancel a job rather than letting that run to completion. If you made a mistake, it's not going to be important. Okay, I'm going to refresh that and give it just a few more moments. Okay, it's completed. So what I'm going to do is click on the name of the job, and click on the name of the job will actually take me to the output folder, exactly where the results are placed. And I can click on the results to view them. And there are my aligned files. Okay, so you can definitely navigate quite easily through any of this to be able to see what you did. Okay, this worked the way it was supposed to. There's another way to relaunch the app. So if you click on the app name here, it will relaunch, and then it will have your, in this case, the same parameters that I had used previously. Does that work as well for the other one? Yeah, okay, so it has everything there. All the input files I used that time, and I could go back and say, oh, you know what, I really want this alignments to be, 10 instead of 20. Okay, so I think you get the idea. We're going to have to move on from the discovery environment to our next platform. Again, if you do have any questions, please, at the bottom there, go ahead and chat me those questions. I'm more than happy to answer. We've got only about 10 minutes left, but I think it's enough to show you how to get started with Atmosphere. Okay, so back to the slides. One last comment, if you want to see a complete listing of all the applications, back on the slides here, I have a link, and you can also look in the demo documentation at iplantc.org/apps1. You can see a complete listing. Okay, so we did the demo, we did the alignment, and now we're going to move on to Atmosphere. Now, Atmosphere is a cloud compute system, and you may not be too familiar with cloud computing systems, but you will, let me give you a little bit of an introduction. Now, I see a question that's coming in, so let me take a second to answer it. It looks like it's not finished. So maybe, Dan, if you finish answering for applications like Muscle that have numerous user-defined parameter settings, but you didn't finish it, so I'm going to wait till you finish it, and then I'm going to come back to it and see how I can respond to you for that, okay? In 1 second, let me adjust this here, I have more window. So if you can finish that question, I'll be more than happy to answer it and come back to it. So back to Atmosphere. Let me give you a little bit of an introduction by first laying out what is cloud computing. So the analogy I like to use is, what if you bought a new computer and you want to move all your files from the old computer onto the new computer? Well, you could take your files and your folders, images, movies, whatever, and you can move them onto a hard drive, an external drive, and then those are your backups, and now you can move them onto the new computer. But there's another type of file that you can make, which is called an image file. And with an image file, it is a file, but it's everything on your system, including the operating system, including all your settings. So you're probably used to, when you buy a new computer, you have to reinstall certain programs, you have to update them, you have to put your desktop the way that it was. But with an image file, everything is the same. So you can take that new computer, documents you copied from that external hard drive will still be the same old documents, but the image file will be a complete clone, the files, the data, everything for your new system. So that's, that concept, that image, is going to be important. So remember that word, image. We're going to use that in a second. The next concept is instance. And what does that mean? Well, in the iPlant cloud, we have a whole bunch of disks, CPUs, memory, RAM, all of those different things that you are able to access. And when you want an instance, what you're doing is you're asking us for a little bit of these. So you tell us how much disk space, how many CPUs, how much RAM. And then you're also going to take an image, which you select from our catalog, or eventually you can create your own, and then you make them into an instance, which is a virtual machine. This is a computer that's for your use, residing at iPlant, which you can do anything with, and it's a virtual machine. So what can you do? Well, first let me just talk about the Atmosphere system, and I'll give you some examples. But with Atmosphere instance, you can choose an existing image, you can customize that. You can get a machine which is up to 16 cores or 128 gigabytes of RAM, which, if to buy that off the shelf, would be several thousands of dollars, maybe more than 10,000, I would gather. You can access it via the shell or VNC, which we'll show you in a moment, and you can share your images with selected users or even make them public. Now, there are a couple of use cases for this. I've just outlined a few, but if you have software and data that you're running on your laptop, and you can run nothing but your software and data on your laptop, because when that analysis is going, you lose that computer until it's done, well, you can run those things in Atmosphere. You can also use Atmosphere for visually oriented programs. So a bioinformatics app that already has its own interface, you can't use in the discovery environment, but you can use in Atmosphere. If it's an open source program, you can install it and get it working there. You also have sudo access, so you don't need an administrator. You can definitely install and configure everything at will. It's really great for teaching or for a uniform lab setup. And also, if you're making software scripts available to the community, you can install them and develop your own instance. So I'm going to show you in a demo essentially how to connect to an Atmosphere instance. There are several ways. All of the instances you can connect to via the command line, via SSH, using either PuTTY or your terminal. And many of the instances which have a desktop can actually be accessed, as we're going to do, through VNC. Okay, so let's do that demo really quickly. Come back to the iPlant Collaborative homepage. Okay, and this time what we need to do is, where it says Atmosphere, you're going to go ahead and click on Launch. All right, now I already have some things running, which I'm going to show you in a moment, but what you'll do is you'll click Launch New Instance. And then you'll first see a collection of images that you can choose from to work with. So I'll give it a second for those to load. Okay, so there are a bunch of featured images. If you scroll through, you may see tags to the images that give you ideas of what might be available. I can see stacks, maker, image processing toolkits. I can see tags for phylogenetics, for BioPy, NumPy, genome annotation, etc. So you can really search through and see what is of interest to you. I'm going to go ahead, if I want to launch something, I could then rename my image according to what I want, and then I could choose the size. The caveat is, of course, not every single instance size is available at every single second. There is also some allocation, so you can see how much you'll be allowed to run based upon what you've run so far this month. And of course, if you need more resources, you can request more resources, but your allocation will be refreshed at each month. In fact, before you even come into Atmosphere, if anybody's trying now, you may see you don't have access. Let me remind you from the iPlant homepage that when you come to iPlant, you don't automatically have access to Atmosphere. So you're going to go to the user portal from the homepage where you created your account and log in. And then from there, you're going to see Atmosphere, except it won't be under My Services as mine is because I have access. It'll be towards the bottom under Available, and you can click Request Access and fill out the form. You must be registered with a .edu or .org email account. Okay, so back to Atmosphere. Once I select what I need in terms of those instances and my size, I would then click Launch Instance, and within a few minutes, I'm going to get an instance over on the left-hand side. When that instance reports that it's active and the color is green, I'm then able to connect. So I've already downloaded VNC Viewer, so I'm going to use that. But there's also, if I click on VNC, in fact, let's do it that way. If I click on here and say Access via VNC, it'll launch a Java application. So you could either download it, or if you have Java, that'll work fine. Okay, I don't need the update right now. Skip that, run that, and connect. I'm going to have to continue and then click enter my iPlant username and my iPlant password. Click OK. And now I'm connected to my virtual computer. This is a virtual desktop that is running, and it's just like another laptop or another server that I would have running somewhere. I could do any type of analysis on it that I'd be interested in doing. Okay, if I wanted to connect to this via the shell, via SSH, I can do that as well. Let me start a new terminal. Excuse me. And all I'll need is my IP address, which is back here. And make it a little bit bigger, and then I can do SSH, my iPlant username at my IP address, and hit enter. And I can say yes to continue, enter my credentials, and now I'm connected. All right, if I want to, for example, create a directory, and you should be able to see that on the left-hand side that directory is there. So I'm connected two different ways. If you do download VNC or you have that installed, you connect with the same IP address. You're just going to have to enter :1 after it. Connect. It's really the same thing on that standalone program. I don't know how valuable it is outside of the educational purpose, but you can see I can connect in a variety of ways to that remote computer. I could even share that computer with somebody else so they could see my desktop and work with it. Okay, I have a question from Brian about .gov registrants being allowed to access Atmosphere. I believe you should be able to, so just go ahead and email support@iplantcollaborative.org. So we do have a lot of users who are with the government, and they also have access to it, so you should be okay. Okay, all right, so coming back to Atmosphere, the last thing to know is that as you're running something on Atmosphere, it is going to spend down your allocation. Again, if you don't request more resources before your allocation is through, your instance will be shut off until you're refreshed. You can pause or stop an instance when you're not running it. And of course, once you're completely done with an instance, you're going to want to click Terminate. Now be careful, you want to transfer data off of that instance before, because once you terminate, you will not be able to recover anything. No one will be able to recover anything from that instance. So what could you do on that instance? You could go ahead and start Cyberduck on that instance. You could go ahead and use iCommands and transfer the data back into the regular iPlant data store. You go back to the iPlant Collaborative homepage, there's another tool at the bottom called Ask iPlant. And this is really the best way to answer your support questions. You can go ahead and post your question anytime, and then other members of the community and even iPlant staff will go ahead and answer your questions for you. So we encourage you to do that. In fact, there are a number of things, support vehicles. The Ask That forum is certainly one of them. But also we have plenty in the Learning Center of Getting Started guide, tutorials, videos, documentation. We're always doing workshops, so you can come to your venue in some cases and actually do a full-on workshop. Webinars are also a great way for us to reach our community. And finally, a thank you goes out to all the people who make iPlant possible, including our executive team and many, but certainly not all, of the staff, students, and faculty who have contributed to iPlant.